So hi everyone, welcome to the fourth seminar series for Spatial Omics. And this is really an exciting time. And as we are getting closer to the end of 2020 and vaccines are coming into play, now today we are excited to have our first speaker, Dr. Roy Wollman, or her professor. And I'll be quickly giving you the biography of him. And Roy received a Bachelor's of Science in Biology and Statistics and many others from Tel Aviv University in Israel in 2003. And then as a graduate student at UC Davis, Roy worked with physicist Alex Mogilner, a pioneer of quantitative biology, on several fundamental problems relating to cell polarity and mitosis. And he received his PhD in cell and developmental biology in 2008. Then he, in his PhD, worked with a lot of microscopes, software development, including Micromanager. And I also used to use it a lot, not nowadays, but yeah. And it's a pretty cool custom software. And then he contributed to the several experimental, theoretical, and computer modeling studies. And indeed, his broad expertise in imaging formed teaching at the marine biology laboratories. In 2008, he joined Dr. Tobias Mayer's laboratory as an NIH NSF fellow, postdoctoral fellow at Stanford, where he discovered the mechanisms of coordinated oscillation in cortical actin and calcium that explain conflicting data in the field. And until 2016, he was a professor at UC San Diego, and in the Department of Chemistry and Biochemistry, and later in the Department of Developmental and Cell Biology. And after 2016, he has become a faculty at UCLA, where he currently conducts spatial omics related work, and he's been applying cool modeling approaches on spatial omics, a spatial MERFISH type data or sequential fish type of data. Without further ado, I'll turn to Roy, please.
Awesome, thank you very much, and thank you for introduction, and really thank you for organizing it. As I said earlier, it was really been one of the highlight of 2020 for me, which is not an easy year otherwise. Okay, so today I want to share with you guys some of our work sort of over the years. It's really trying to bring a lot of concepts together, really trying to ask questions about what are cells and why are sort of, kind of I phrase it as, why is not all cells the same, right? So really trying to ask cellular heterogeneity, cellular state, and what are those things kind of connected. Okay, so let's get going. So cells are the fundamental building block of tissues, right? If I think of this Lego frog here, right, it's really built of a lot of small building blocks, right? Lego in this case. And if you would have dissected it up and open it, right, you'll really see all the organization inside, right? All the different sort of tissues and organs, right? All of there. And each tissue is really a collection of those multiple, in the case of this Lego frog, identical blocks, right? That together enable the function, right, of whatever organ or tissue we're talking about, right? But when I say that, right, sort of I always feel the sort of heavy weight of Boltzmann on my shoulder, because nothing is really identical, right? So trying to say that I have identical Lego blocks, I mean, maybe it's true for Lego, and it's actually amazing how good Lego sort of blocks are reproducible, right? The precision there is really amazing. But in biology, right, cells, those building blocks are not really the same, right? There's always some variability around them. So it's really that variability that's kind of the driving question for us, right? Trying to understand what is really going on behind them, right? How can you build sort of complicated organism, right, when you're actually not necessarily using the most precise building blocks, right? So what really causes cellular variability, right? Why are those things not identical to each other, right? So you can broadly think of two sort of extreme answers to this question. On one side, it's just noise, right? So, you know, biology is complicated, and for a lot of genes, right, you have only one copy of, or two copies of DNA. Sometimes only one of them is active. Small numbers, it will just generate noise, right? So it could be that the reason we have variability is purely that, right? We just you know, unlike Lego blocks, right? It's harder to create cells that are really identical and really, really precise. But then the other explanation is more of a sort of deterministic variability, right? So if I would look at a sort of football game, I will say there's a lot of heterogeneity in football players, right? I mean, sure, they all have those helmets and big thing, but some are big and some are small, right? And as an unexpert, which I definitely am when it comes to football, I will really can say, oh, look, there's a lot of variability among players, right? But really that variability can very much be explained if I know the role of those guy in part of the game, right? So the, yeah, here's where I show my ignorance in football. The sort of wide receivers, right, will be sort of smaller comparing to the sort of whatever guards, right? So there's always this, even though if I don't know their roles, right, if I don't know what they're doing, I can interpret it as a big variability, but it's really, there's a sort of hidden variable there, right? The role, the functional role of that player that can really explain a lot of what I would otherwise consider to be sort of, you know, just pure variability, right? So those are maybe too extreme, right? One of them is that there's just noise, right? It's pure stochasticity that's uncontrolled. And the other case, it's much more regulated, right? It may be trying to argue that there is sort of task allocation, or really sort of some specialization within your population of football player or cells in our case. So I really like this picture because it's sort of, you know, kind of when I was a graduate student, that was the kind of the key image, right, visual of what system biology was at sort of early 2000-ish. And it's nice that you can now buy it as a sort of car magnet for only $8.99. And I will guess that in the future, right, there will be car magnets sold on something that looks kind of like that, right? Sort of this a version of this type of plot will be sort of our era, sort of kind of call for arms, right? Of some picture of really trying to capture the essence of what's going on here. Maybe sort of kind of similar to this yeast protein to hybrid interaction. And right, I mean, I'm sure for this audience, I'm sure all of you understand what I'm showing, right? This is just a t-SNE plot of all the sort of single cell RNA-seq of a whole bunch of cells from a mouse. And this is really trying to depict some sort of space, right? I mean, to the projection of some complicated gene expression space that really says that a lot of cells are sort of in there. And really that idea, right, that cells are sort of somehow in a cell state space, right, or cell type space, right, and depending on the resolution that you look at it, right, is really the driving question here, right? It's really trying to say, is this, how are those sort of different roles that cells are playing, right, are acting, and not when you're talking about sort of far away population, but even when you look at very close cells, right, cells that are clones or for the same cell type, are there still sort of differentiation and roles that they can play within them? So in this vein, I'm going to talk about sort of three interrelated stories, right? The first will be really trying to examine this concept of cell state space a bit more and really try to argue that we can maybe even bring some mechanistic views into that thinking. In the second story that I'll share with you guys, it will really try to answer a question about the precision in those space, right? How sort of, if I have a point here, right, how accurate it is, right? Sort of maybe related to this question of, right, what is the, how much noise do we actually have in the system, right, when we talk about heterogeneity. And finally, we'll get to the point that, you know, would bring us all together is this try to ask, how do those states, different states of cells really manifest themselves if we actually know where they are in sort of, you know, geometrical space, right? What is the connection between this abstract space of gene expression, right, where we're sort of talking about cells are sort of spread into some abstract gene expression space, and actual geometrical space, right? What's the connection between them, and what can we learn when we bring those two things together? Okay, so a big hero of this talk today for the first sort of two part of the talk will be calcium signaling. I love calcium signaling for a few reasons, but among them is that it's really one of the most studied signaling networks. So I always feel that we're working in an area that a lot is known about it. It's a very fast signaling response. So within 10 minutes or so, right, from the addition of ligand until the whole thing is done, it's, you know, roughly 10 minutes, depending, of course, on the details of the cell line and the ligand. But overall, it's a very fast signaling response. And basically, it's there in every cell. So this specific pathway I'm showing here, the ATP binding to GPCR, is basically active in all cells in your body as part of the sort of a damage sensing and wound response mechanism. And as I was saying, right, it's really very well studied, and there are even sort of textbooks about mathematical models of calcium signaling, right? So it's really one of those places where we can really leverage a lot of prior work that was done to sort of capture what's happening inside that signaling pathway, right? And sort of just a few words just to sort of put everything in context. We have a sort of in calcium signaling, right? The GPCR gets activated, activates phospholipase, generating IP3, releasing calcium from the ER, right, for IP3 receptor to the cytoplasm that are then being pumped back into the ER, and that's basically what's caused this sort of spike of calcium that we're seeing in this GPCR activity. There's usually a main spike, and sometimes there are a few more spikes. But again, this is sort of depending a little bit on the experimental system. Okay, so the sort of what we brought here, right? The trick that sort of we're using here are to help us examine calcium signaling better is really trying to combine this live cell imaging of calcium biosensor with sort of MERFISH, right? With the ability to actually ask what's happening inside the cell. So this is sort of a quick video of a few cells, right? And we can do our standard sort of cell segmentation and tracking and measuring calcium dynamic for this. Those cells express a GCAMP calcium biosensor. And then on the cover slip, we have some fiduciary grids that we can then use sort of to align. And the same cover slip then goes for MERFISH imaging. So we can then image sort of lots of the expression of lots of genes. And we chose those genes such that they basically cover everything in the calcium signaling network, right? And just to show you, right, then we, in the end, we sort of align those two things. So really what's unique about this data set is that we have both the live measurement, right? We have sort of for every cell, we have its calcium response trace, and we also have the vector of the sort of MERFISH, right? The gene expression of roughly 150 genes that we chose to really represent what's happening in that signaling network, right? And again, for this audience, I don't think I need to explain what MERFISH is, but just in a few words, it's this sort of beautiful idea that says that we can sort of hybridize multiple colors to an RNA molecule and use some sort of barcoding scheme to really tell us later on this spot, right? What was the identity of this spot in terms of what gene it was? And it took us actually quite a few years to get MERFISH going in the lab. We started around, or still in San Diego. So it took roughly 4 years to get it working. But now it is, so we're very happy about it. So for this specific data set we're going to talk about, we measured roughly 150 genes related to calcium signaling, including a few cell cycle markers and a few other sort of markers related to this cell line. So those are MCF10A cells, or sort of epithelial cell line and epithelial breast cell line, and sort of they go for a little bit of differentiation between lumen and basal. So we had a few markers there, the cell cycle genes, and then most of the genes were basically genes related to calcium signaling. And so mentioning what's nice about calcium signaling that it is fast. So by basically we can assume that no RNAs are changing during the time of sort of signaling. So even when we fix the sample after 10 minutes, and the RNAs we're measuring are basically the RNA that were in the cells when sort of the ligand was added, right? There wasn't any change in those 10 minutes, right? Really allowing us to say that sort of these are the RNAs of the protein that are actually in charge of whatever is happening in the cell, right? Of the signaling response itself. Okay, so now we're going to, okay, we have those calcium signaling and we have this unique sort of paired data set. What will be really a good way to represent what's happening in the cell, right? So maybe the most straightforward thing is we have those live cell measurements, right? We know what calcium dynamic is in response to a ligand. So this is maybe how it sort of looks on a full well, right? You see all those, the response of calcium in all those cells, right? There is this big thing, and on top of that, there is some sort of blinking. And we can do our sort of standard image analysis. So here I'm showing you just sort of a subsample of those cells, either as a heat map or just as a sort of trace. They're, you know, right, just over time. So you see the sort of the calcium response over time, right? And the whole thing is a little bit more than 10 minutes, right? We add the ATP roughly after 3 minutes. So basically when the spike goes up. Okay, so this sort of time series, right? It's actually one representation of what's happening in the cell, right? You can think of it, if you will, as a phenotype, right? Because that's sort of the end result of the gene expression, right? What the cell is, right? It's such that it can respond to a ligand in a specific way. So this is sort of the response. So, I mean, it's a phenotype. You can also say that it's intermediate, right? Like exactly what you call a phenotype is always a relative thing, but it's sort of downstream of a lot of things, right? That's a result of the ligand and the results of what the cell has in terms of its expression and its construction and sort of overall unit, right? So this is one way to think about cell state space, right? We have lots of cells, each one responds to calcium differently. So we can really use sort of the phenotype as some interesting sort of readout that tells us something about the cell. Right now, another option, which is maybe what we're more familiar with thinking about when you think about cell space related to those sort of Tisney diagrams I alluded in the beginning, is really think of the transcription signature in the cell, right? The gene expression in the cell, right? That's what we all do when we do single cell RNA-seq and similar methods like that, right? And I'm sort of showing you just the, right? We can take all our genes, we can do sort of gene-gene correlation matrix or all the sort of standard statistical analysis of those type of big data sets. And we can potentially cluster those genes into sort of different clustering. So this is sort of Levin clustering, and I'm just showing you the over those from those 5,000 or so cells that we measured in this experiment. And what was very sort of gratifying, even in the first pass, is to see that there was really a strong correspondence between the sort of state of the cell when we look at its gene expression compared to what's happening when in the calcium, the phenotype level, right? When we actually look at sort of calcium response. So these are the different sort of clusters that I showed in the previous slide, sort of just displayed differently. And then sort of the cell in that specific cluster are in black dots in the gene. And we also came up with a sort of a two-dimensional representation of calcium signaling. And it's really nice to see how sort of cells that are close to each other in gene space are also close to each other in this phenotype space, right, in the calcium trajectory space. And there's a pretty good correspondence between those two spaces, really arguing that, right, sort of the state of the cell can be manifested, so it's manifested in two different views, right? One view will be the sort of gene expression state of the cell, whereas the other is what is actually the cell is doing, right? Its phenotype in terms of its calcium signaling. And what's very nice is that we can now go drill down and actually look at the specifics. So those are sort of 5 of those of the many genes that are related to calcium network. Those are the 5 pumps. So sort of our expression of a few of those key pumps. So 2 circa, so the pump that pumps sort of back to the ER, PMCA, the pump that pumps from the cytosol out to the plasma membrane, and another pump that pumps calcium into the Golgi. So you can really see how those 5 pumps are really distinct from each other according to the sort of prescriptional state of the cell, right? So for example, in this cluster here, right, all those pumps are really strongly expressed compared to sort of the cluster here where they're all sort of lowly expressed, right? And if we can then, because we have the calcium measurement for each one of those cells, we can really see that in when the pumps are really highly expressed down here, right, in sort of cluster 0 here, you really see very weak response, right? There's sort of small peak and there's not much afterwards. And you sort of see that both in the live sort of traces here that I'm showing on the left, and we're sort of just measuring the peak and the width of that first main peak, just as sort of 2 statistics to characterize this sort of continuous time series. Right, so it's really nice to see that there's really correspondence between gene expression space and calcium signaling space, right? Really arguing that maybe we can start learning things from back and forth between them. Really, how does those different genes, right, then the protein that they express, really sort of shape the details of calcium signaling response? But then I'm going to argue here that maybe we can bring a third view to this problem, right? Another way that we can think about what's happening inside those cells that maybe can really help us look at things a little differently. And really what I'm arguing here is that we want to bring in this, everything I showed so far didn't actually use the sort of many years of really good biochemistry and cell biology that was done on calcium signaling. And we're trying to think, how can we bring all that sort of knowledge, right? There is a lot of knowledge here. And how can we leverage that to get a better sense of what's going on inside the cells? And of course, one way to do that is to really think of, right, the sort of mathematical model to represent calcium signaling, because mathematical models are nice, right? They are actually capturing a lot of information, right? And you have to also do some trade-off, like what how detailed you make your model and what you capture and what you not capture. But they're really sort of comprehensive views of what you think is a good representation of the signaling in that cell. Right, so we can sort of take those kind of another version similar to what I show you, maybe a little bit more detailed, and really construct sort of kinetics of all the different reactions in those cells. And each one of them, I'm not showing you all the equation, I'm just showing you sort of one of them, just how the IP3 is changing. And we can write the mathematical description of what's really happening there, right? ATP binds to the ligand, and et cetera, right? I'm not going to go for the entire detail of the math, but hopefully you get the sort of gist, right? Nothing here is sort of fancy. This is just simple sort of Michaelis-Menten and simple sort of kinetics, right? Nothing too out of the ordinary. All right, we're very much leveraging all the sort of lot of work that was done on mathematical modeling in calcium. So this is sort of not our model, right? This is sort of a known model of calcium signaling. And in our model, we have sort of 17 different parameters, right, that really describe what's actually happening there, right? So the reactions are the same for all cells, but sort of the hypothesis or the reason we're bringing those mathematical modeling is to say, well, maybe what I can think of those parameters really as the sort of tuning states, right? So those the signature of those 17 parameters really, right, can describe together with the ligand that I'm adding what will be the response of the cell, right? Because the structure of the network is potentially shared across all genes, right? The model is true for all cells, right? It's the same model in all cells, but what's different between different cells, right, could be that they're just the parameters, the details, the specific parameters of what counts of that model in that cell could be a bit different from each other. And kind of our thinking was that maybe this is another representation of what's actually happening in a cell, and maybe that's a representation. It will be more informative because it does leverage everything we know about calcium signaling. It does leverage all those sort of painstaking years of biochemistry and cell biology that sort of were used to figure out all those different reactions, right? We're not sort of starting de novo, and there's no reason we should sort of pretend that we are. Okay, but then of course, what we're missing here is the ability to tell every cell what are your sort of specific parameters in that cell, right? So that's our next task, is to do this sort of parameter fitting, right, for every individual cells, and and that's what I want to describe in the next few slides. So we're doing that in a probabilistic framework, and the reason we chose that is we really not only care about what are the parameters in the cell, we also really wanted to get a sense of our confidence in that parameter, right? What is how how accurate do we believe we can actually predict what the parameter, the specific parameter value in every cells are? So in some sense, we're not just looking for the best fitted parameter, we're really trying to find a probability distribution, right? If you will, the posterior of the parameters given the data, right? So how do we get that, right? How can we sort of estimate that probability? So our answer to this question was to use sort of a statistical framework called Approximate Bayesian computation, which is really sort of a rejection sampling on steroids. So it's rejection sampling with a few sort of kind of twist around it to make it sort of much more efficient. Right, so what's sort of the core idea behind it, right? This is what we're trying to do, right? We're trying to learn this type of distribution, right? What are the potential values of those parameters given the specific data that we have about calcium in one cell, right? So we'll repeat this procedure for every cell that we're looking at. So first time we'll just say, okay, I know very little. We'll just make a guess of what are those possible parameters, and our first prior is very, very broad, right? We're sort of few orders of magnitude. We're allowing the parameters to vary by few orders of magnitude every time. Right, but then we can sort of randomly sample from this prior, right, and get a bunch of candidates, possible parameter values for this specific cell, and now we can look at them. So this candidate actually generates a pretty good fit, right? So awesome, let's save this, right? This candidate generates an okay fit. Sure, I can maybe still save it, right? It's not that horrible. And those candidates are pretty bad, right? So I probably don't want to save them, right? So I'll sort of remove those. And that already tells me that I should have a lower probability density in the outside and sort of higher in. So already I'm kind of updating, right, my sort of, kind of estimate of the probability density in that cell. And then sort of the trick comes that we now do the whole thing sort of over and over and over again, right? So we start with this, we sample those, estimate our kind of posterior, and then basically use it again as our new prior, right? And we can sort of keep on doing that lots and lots of times, right? And potentially tens and hundreds of iterations until in the end we get a pretty nice sort of estimate of our probability density, right? Or in other words, what are those parameters, or sort of probability density of those parameters in one given cell, right? So let me show you sort of for 3 individual cells, some sort of the actual experimental data, or which we smoothed a little bit to make the fitting a bit easier, is shown here in the red dots for those 3 cells. And then sort of we allowed, because we have this experimental uncertainty, we also allowed the fitting to be up to some error bars that are determined by how good we actually know that experimental data, right? So this is sort of our 3 good fits of sort of data, or sort of families of fits, right? Because we're allowing multiple parameters in the end to exist, to sort of to survive our sort of rejection sampling, right? Our procedure. So those are for the all sort of sample of trajectories. We're all considering sort of good fit for sort of cell 1, cell 2, and cell 3. And if we look at their sort of distribution of those 17 parameters, and I'm going to show you those parameters, sort of those are the different 17 parameters, and sort of we normalize them to a reference, and then to log 10 of that. So really, it's sort of just a log 10 of the value that you're getting over the what we sort of initially guessed, or sort of literature value, if you will, of what was in there. So the first thing I want to point out is that look how uncertain we are, right? So we do have a lot of uncertainty here, right? So there's sort of, in some cases, an order of magnitude, and in some cases even more than an order of magnitude uncertainty. But what are the specific value of this parameter? I mean, some parameters we're a bit more certain about, some parameters we're less. But overall, I will not try to claim that we know what those parameters are exactly in every individual cell. But on the other hand, what was very nice is to see that sort of if we even though we have uncertainty, it's not the same in different cells, right? So there are definitely changes, right, between, so in these two cells that are more close to each other, the distributions are actually more similar compared to sort of, if I look at like the fourth one here, right, or right, like there's definitely differences between those different sort of distribution if you look at them. So we can use those matching between those distributions as a way to estimate how similar two cells are. And again, what's nice about it is because we have the sort of uncertainty, we can take that into account. So it's not just how different two cells are, it's how different they are compared to how sort of certain we are in those parameter values, right? So using this sort of a distance metric, right, the KL divergence between those probability distribution, we can now look at all those cells again, right, now through the eye of those parameters, right? Of those, the signature of the cell now is the 17 parameters, right, that we are inferring about what is the, right, which we need to parameterize this sort of kinetic model of what calcium signaling is doing in this specific cell. And what was really interesting about this exercise and what really popped out is that there are really very distinct clusters of cells in our population that were not so easy to see when we only looked at calcium response, right? Calcium response seemed to be more or less sort of continuous. Sure, there are some cells that are sort of weak response, some cells that are large, sort of have a high peak response, but it was very hard to sort of see what's happening there when we only looked at the phenotype. But now when we interpret this phenotype through the lens of the mechanistic model, what could explain such a specific pattern of like a big spike or a small spike, right? There are actually very distinct parameter regimes that are needed to explain those things, right? And they're really sort of the biggest difference between cells that are sort of doing very, very little to cells that are responding somehow. But even the cells that are responding somehow, there's sort of the weaker response and a bit more stronger response among them, right? So there's really sort of 2 to 3 sort of clusters or sort of distinct cell states in our population of, clonal cells identical from the same, MCF7 cell type, right? So really arguing that bringing this mechanistic view, right, allowed us now, if you will, just find a new lower dimensional representation, right? It's kind of like almost like a mechanistic type t-SNE, right? When we're reducing this 300 points of calcium signaling to another signature of 17 parameters, but now that signature is really informative about what's really happening there. And what's nice about this is now those parameters, each one of them is not some abstract dim1, dim2. Each one of them is really a specific statement about what's happening in those cells, right? Each one of them has a label in terms of what is actually happening there in the kinetic of the cell, right? And one of those key differences in parameters really pointed out to this IPV receptor, right? Arguing that that sort of the key difference in parameters between our clusters was the concentration of IPV receptor. So just to test this idea as a prediction, we did the following experiment, right? We estimated, we looked at ATP stimulation, so the same thing I showed you earlier, and kept on tracking the same cell, and now on the same time did it IP3 uncaging. So we're sort of, rather than stimulating the cell from the outside, we sort of loaded cage IP3 and sort of tickled the cell sort of in internal node in a network, right? And we can sort of measure those 2 spikes, those calcium response in the same cell, sort of separate from each other by sort of an hour to let the cell recover. Right, and what was very nice to see is that sort of exactly as our model sort of analysis predicted, right, knowing the response to IP3 really explains sort of 70% of the variance in just what the response to ATP will be, right? Really pointing out that sort of the kind of a representation of cells, right, in this sort of parametric or mechanistic view of cells really pointed out what are the actual differences between those cells, sort of in terms of what part of the pathway is really the one that's sort of distinct among them. Right, so kind of the gist here is that sort of single cell model fitting, right, could really provide a useful and sort of mechanistically driven way to sort of represent the cell state space in a way that I really think of it as a very complementary to sort of its representation that we're more kind of comfortable or thinking more about in terms of its gene expression space, or sort of just looking at the phenotype itself, right? And those 3 are kind of complementary views to each other. What we're still doing now, and it's a collaboration we started with Adam McLean from USC, is really trying to ask, can we sort of really fill up the circle? Sort of, are those gene expression states, how do they correspond to the specific parameter value that we're inferring in those cells? And there's definitely some strong correlations, but we're still too early for me to give a full answer of what are those correspondence. But I think it will be really interesting to see, can we actually represent those 150 calcium genes in those parameters, right? The expression of which genes really changes what aspects of calcium signaling. And because our sort of representation doesn't include a parameter for every gene, where I'm expecting a few to sort of map to the same thing, which will really provide a nice way to go back and forth between sort of mechanistic view of calcium signaling and what are the genes that are driving each one of those parameters. Right, so kind of the argument I was making in this sort of vignette is that there is sort of this cell state space, and we're really sort of the classical kind of parallel of the blind man and the elephant, right? We're all looking at the cell state space in different sort of angles, right, in different ways, right? And I sort of described 3 of those today. One of them is just looking at the phenotype directly, right? So it's sort of dynamic signaling response of calcium, right, to our ligand. We're also looking at it for its gene expression state, right? So it's another view that we can have over basically the same entity, right? The same cell state space. So it manifests itself in different ways. And of course, what we're trying to add to this is actually a more mechanistic view, right? It will also basically allow us to really interpret and understand what those cells are doing, right? And what's the sort of variability between them and how we can mechanistically represent what's happening between them. Okay, so with that, I want to talk to sort of move to the next question I was posing, which is more related to noise, right, and how noise plays here. And sort of my, it was work by previous grad student Rong Fan, and he really phrased it nicely as sort of the turtle problem. So why do we think of it as a turtle problem? So right in those sort of t-SNE, right, those cell space representation of the gene expression space and the cells, right, you sort of take your cells and you sort of divide them into different sort of region, right, according to the cell type. Now, what was curious for us is we wanted to say, okay, let's say that my measurement was perfect, and I took one of those clusters. I took the whatever cell type was represented by red cells, and I redid the same measurement, and I redid t-SNE. Will I can break those cell type now into lots of new clusters, right? Because there's sort of more sub-differentiation within the cell type, right? I mean, at some point, right, this exercise should stop, and it sort of reminded him this idea of sort of, right, turtles under Earth, right? Are there turtles all the way down? Like at some point, right, 2 dots in that space, we should consider them identical or sort of similar to each other. And our logic was that what really determines that is noise, right? So, right, like any other type of measurement, what really determines your resolution is your sort of noise in your measurement, right? If the noise, or not really measurement, right? Noise in terms of how cell can control that value, right? Maybe I should say it's not just measurement noise, it's really a biological entity, right? So if a cell cannot control its position in that space more than some sort of small radius, right? I should not really, right then I should not consider those 2 cells sort of different from each other, right? They're kind of within noise, right? Whereas if, right, the noise is really small, it will matter. So really understanding the gene expression noise, we argue, is really fundamental for us to understand sort of what is really the resolution that we can expect to get in this sort of cell state space that we're sort of interested in. Okay, so we're trying to think, okay, what could cause noise in gene expression, right? What are the 2 main sources of noise? And of course, there's sort of beautiful work really describing that sort of gene expression noise can really be sort of decoupled or deconstructed into 2 components, right? Sort of a cis noise, so things that are happening on the promoter itself, right, or in the gene itself, right? So, and of course, trans noise, right? So concentration of transcription factor, the cell size, the cell cycle, everything that's sort of shared among multiple sort of transcription factors, right, or multiple genes, right, we'll consider sort of trans noise of source, or some people call it extrinsic, and everything that's sort of unique to this specific gene or this specific allele, if we're more exact, right, will be considered sort of cis noise. Right, and there's a really nice, because of this idea that sort of trans noise is shared and cis noise is not, we can do this nice decomposition of sort of noise into, or sort of look at the covariance between them and really see how informative it is, right? So this is just a sort of illustrative cartoon, right? This is not measurement, this is just a sort of simulated data, just to make the point that if we take those kind of just the measurement themselves of gene A and gene B, right? If they're sort of really cell state dominated, we're expecting to get a pretty high correlation between them, right? So this is sort of in the red here is the correlation value, 0.82. And if they're not, right, if they're sort of intrinsic dominated, we can still get a little bit of correlation because of the shared cell state, but the correlation value we expect to be lower. The second thing that's interesting here is we can also look at the Fan factor, right? We can also just look at the sort of dispersion, right? How much variance we're getting as a function of the mean, right? And now what was nice is if indeed, right, we can do this sort of interesting procedure, we say, okay, but what happens if I know one of those things? What happens if I know the concentration of, let's say, transcription factor 1, right? What will happen, right? And I condition my measurement rather than looking at the raw data, I can look at the raw data and regress out my knowledge about the concentration of transcription factor 1 in each one of those cells. right? If we're intrinsically dominated, not much would happen. Sure, the correlation will go down a little bit because I removed something that sort of caused correlation. And the Fano factor might reduce a little bit because, of course, I'm regressing out some factor that induced variance, but not by much, right? And if I keep on doing that all the way to sort of distribution factor 2, I should get to a point when there is no correlation anymore between cells because they sort of regressed out everything that is shared between them. And then really what I have left there is just an estimate of the dispersion, right? The Fano factor. Now, if it's really cell state dominated, right? If most of my noise is in sort of trans noise, right? As I do this procedure, as I sort of condition or regress away the effect of distribution factor 1, right? The correlation go down, and also the Fano factor goes down potentially more than it went down in the sort of extrinsic noise dominated. And if I do it all the way, if I actually correct for all those things that are sort of shared among cells, right, you should get the correlation 0. And ideally at that point, you'll also get Fano factor of 1, really arguing that what you get is just sort of Poisson noise, right? Because there's really not a lot of intrinsic noise in that cell, and Poisson noise is kind of our limit, right? There's sort of a floor noise, sort of noise of how not sort of, we're not expecting expression to ever be down less than sort of Fano factor of 1. Okay, so what are we sort of, we don't actually have the measurement of those distribution factor 1 and 2 in our case, but we do know calcium signaling, which potentially captures a lot of the information that happens in the cell, right? It does capture, right, sort of with the argument that we're looking at multiple views of the same sort of point in cell state. Even though those are things are, by no means I'm trying to say that they're causative for the gene, for sort of variability in gene expression, then our logic was maybe they're very correlated with them, and we can still use them in a statistical way just to regress out shared variance among our genes, right? And because all our measurements are coming from this MERFISH measurement, we have sort of multiple measurement of a gene in a same cell. We're just interested to see what's happening to those measurements of the variability of gene expression in those cells, if we sort of take into account those different sort of shared factors among them. So just to say a few things about those shared factors, sort of volume is a big one that was known before us, right? We can explain a big part of variability among cells just by knowing their sort of cytoplasmic, the volume of the cytoplasm in those cells. And we also had a few genes related to cell cycle, so we can sort of account for cell cycle. We accounted for a few, specifically 2 genes related to this differentiation state of those epithelial cells. Are they more basal or luminal? And all our different sort of calcium features that are really coming from those sort of traces that I already showed you. And what was interesting is now we have those 13 sort of kind of dependent variable and sort of lots of independent variable that we're looking at them, lots of dependent variable, but we're looking at them one at a time, and those 13 independent variables in the axis. So for example, if we look at the expression of one of those genes, ATP2A2, right, as we sort of include more factors, right, we can see that sort of it has a pretty good correlation with it. The volume here explained a lot. Cell cycle is not a huge explanatory factor for this specific gene. The differentiations that it wasn't sure, but calcium X does sort of has a pretty good explanatory factor, right? It does explain sort of 11% of the variance for this specific cell. And of course, for a different gene, those values could be different, right? What aspect explain the variance in each one of those genes is specific to each one of those genes, but we only have sort of 13 of those features, right? So it's not sort of a crazy overfitting problem. What was interesting to see is that those features overall do explain sort of roughly 2/3. There is some range, of course, depending on the gene, but they do explain roughly 2/3 of the variance. Volume was the biggest part, right? So sort of knowing the volume of the cell was always the biggest component, or most of the time was the biggest component. Besides cell cycle, there are a subset of genes that really sort of are changing a lot with cell cycle, and in that case, their effect was even bigger than volume. But calcium and sort of differentiation state also were sort of significant contributor to that gene expression variability. But what was really gratifying is when we now did basically the same thing I explained in the cartoon in the beginning, right? We sort of regressed out all those features one at a time and just look at the covariance between the residuals, right? As we explain more and more things, right? What happens to the covariance between the residual? And what was interesting is that we start with a matrix that's sort of full of correlation, or I'm showing you the correlation matrix here. There's a lot of correlation between all those genes, but if you account for volume, all of a sudden, oh, there's less correlation between genes, right? Because we accounted for one of those sort of shared trans factors, right? It sort of correlates among those genes. And as we do more and more of those things, in the end, when we included all our factors, we really got a correlation matrix that was basically all around sort of 0, right? There were very little correlation among those genes. And sort of our argument here is that this really suggests that we have accounted, right, for most of those things that are causing sort of shared variability among genes, right? So we accounted basically for the trans noise, and now we need to ask, okay, how much of the variance is left, right? What is the sort of fan factor of cells after we sort of correct for all those, account for all those additional contributor? And what was interesting to see is that really the so I guess we should start here in the middle. The fan factor for those things, right, as we account for more and more things, really very quickly went down to sort of level of 1. Another way to look at it is just sort of we can do a PCA on the residual and whatever is left after we regress out those key factors. And sort of the first two components in the beginning explain 40% of the variance, but they really go very quickly to sort of very close to sort of permuted data, which is this dashed line. Another way to look at this whole thing, some people like to look at sort of noise as CV square, sort of the variance over mean square. And in that case, right, if you look at CV versus on a log-log plot versus mean, right, a Poisson limit will be sort of a line of exactly -1. And it was really interesting to see that most of the genes we're looking at were basically sitting on that limit, right? So transcriptional bursting or any other sort of gene-specific noise sources seem to have a pretty minor contribution to gene expression variability, right? They're basically just sort of Poisson noise that's hard to control. And most of the variance we're seeing is really coming from some sort of cell state variability, right? Some variability they sort of shared among most of those genes, right? Most likely some upstream regulatory factor or something like that.
Yes, right now I'm in TikTok.
Questions or someone just not muted? Okay. Yeah, feel free to ask questions anytime if you're interested. Okay, so, and with that, I want to sort of move to the sort of last part of it and saying, okay, so we still have this variability and we're right that we're sort of observing, and we really don't think a lot of it is coming from pure noise, right? From sort of inability of cells to control themselves. So what could really explain that? And a lot of my thinking changed once we started to actually move into sort of space, right? To actually look at sort of spatial biology. And this is a beautiful collaboration with the Young Lab and sort of Russell that did a lot of this work, a lot of the computational work that I'm going to describe in the next few slides. So our plan, I'm sure like a lot of, you guys as well, will detect spots, right? Spot is really just for every, right? We have the X, Y, Z and label for each one of our genes. And we were planning to just segment cells, right? And then assign RNAs to those cells. And once we have sort of segmented cells, right, effectively we will have sort of the kind of cells by gene matrix. And then, you can do whatever you want with this. You can take this matrix, classify cells into type, and do all other sort of single cell type of analysis that you want to do on this. And that was our plan. But like all plans, the test of reality was immediate, because when we look at our sort of tissue section, I'm showing you here the mouse hippocampus, which is our tissue that we looked at. I mean, where are the cells, right? Sort of to do MERFISH in this tissue, we cleared everything away. So really there's nothing much. So we still have the nuclei, right? We still have the sort of DAPI stain, but to actually know where the boundaries of the cells are became, right, we just had no information, right? Because really we cleared it all out. And maybe we can sort of, we try to use some background staining of RNAs and so on, but it's kind of, the better your MERFISH is, right, the worse your segmentation signal is, right? So it was really not kind of optimizing MERFISH to get better signal. It really causes segmentation to become harder and harder. So really try to say, maybe we need some other way of doing it, right? Maybe we can somehow change how we think about the segmentation, right? So change the order of operation, right? We kind of, our naive thinking was, we get the data, we segment, and then we convert it into sort of a cell by gene matrix and so on. But maybe there's a better way of doing that. And what really sort of kind of helped us with this is the idea that, again, a lot is known about this problem. So specifically in mouse, right, there's the Allen Institute, and they build this beautiful taxonomy based on sort of SmartSeq and 10x single cell measurement. They have more than 1 million cells, and it did really, a lot of thinking about how to sample those cells and so on, right? So they really build a beautiful sort of taxonomy, right? Single cell data on the gene expression of each one of those cells based on their types, right? And sort of that sort of already a second iteration of that, it just came out in August. And really they represent their taxonomy as a form of just a sort of hierarchical clustering, right? So there's some sort of dendrogram of a little more than 350 different cell types that you expect to see in a brain. And basically you can think of it almost as like a labeled training set, right? Because for every cell type, right, I know it's a label, and I have a lot of examples of the expression, of the gene expression in that cell type, right? So it's really a fantastic label data set for definition of what is a cell type, right? So kind of the way in a way, right, the way that define a cell type is just by giving us examples of what is the expression in cells that have that label, right, of that type. So kind of the computational pipeline we sort of came out of that and is the following, right? Okay, we'll do our best guess and segmentation, right? We'll use the nuclei information and we'll basically try to assign pixels as best as we can to cells. Then we'll take this taxonomy, right? We'll take the brain taxonomy provided to us by the work of the Allen Institute and use those, right, to basically classify each one of our cells into a specific type, right? So we have here in my cartoon example, I have cell of type 2 and cell of type 1. Now at this point, after I did classification, the information of label actually propagated into each individual pixels in my image, right? So now every pixel has assigned a specific type for it, which is my current best guess of what that type should be. And I can use that now to train a neural network, right? Basically to say what is given the local RNA densities, right? So each one of those pixels, right, will have a vector of 80-something different genes that we're using for this purpose of segmentation, right? And we're going to ask, okay, what is the sort of predicted cell type? If this is the RNA expression in this specific pixel, what will we predict the cell type, right? So this classifier is not that different from the overall taxonomy classifier in the sense that they're both converting gene expression into type. It's just happening on the local level, right? This is really expectation of what is the different pixels in the image, right? So locally. So in some sense, we're sort of accounting much more to the variance and sort of sampling issues and everything else. We're really training a pixel level classifier rather than sort of a cell level classifier, right? So now that we have at least one, our best guess at this point of this pixel level classifier, we can reclassify all the different pixels in our images, right, according to their type, right? And as you can see, we don't really think any of them is a type free. And we definitely found that cells that this area that we thought already was type 1 was indeed mostly classified as type 1, and the area that mostly classified as type 2 is type 2. But note here, right, there are a few pixels here that even though we originally guessed them to be of type 2, their gene expression signature in those specific pixels, right, is actually much more closer to type 1 than it is closer to type 2. So now that we know this information, right, we can take into account our sort of type per pixel and do some sort of pixel flipping until we sort of kind of match up better the information of the pixel in its own cell type, such that now we can update our segmentation, right? So now we sort of update our view of what's happening there because now we can include in our segmentation not just our initial guess, but sort of distances and watershed. We can also include in that what's our current sort of understanding of what the, how does the pixel look like for every given type. Now that we have updated segmentation, we just repeat the whole thing over and over again, right? So we take the same taxonomy from Allen, reclassify cells, and now we've sort of basically relabeled our pixels, and we feed them back into the training, and we retrain them again, and we just go over this procedure, lots of times, basically until we converge, right? In other words, we converge where the sort of our neural network is sort of really trained properly, right? In a sense that it really predicts the right type for a given pixel, and sort of you can use that information now to sort of segment cells in our images. Another way to think of this procedure, maybe sort of a bit higher view, is we're really trying to sort of segment cells such that the consistency of sort of cell type marker in our sample is the highest, right? We're really trying to say you are all belong to the same cell because you're sort of more in agreement with each other in terms of what will be the type of that cell, right? So we're sort of not breaking away a specific area, just right, assigning it to another cell if it's sort of more closer to sort of a cell in terms of what are the actual expression in that in those pixels. Okay, and when we do that, right, we can now, what's nice about it is not only get better segmentation, basically in the same procedure, we also get all the different cell types according to that taxonomy of the Allen brains, right? So we get basically for every cell, what do we think is the probability that it belongs to each one of those sort of 350-ish different cell types, right? So that's why I'm showing here sort of in the top left corner in A, and the color code is the color code that sort of Allen people came up with. So we're using their RGB value. They have for every cell type, they have sort of an RGB value definition. And I'm showing you here on the other side just the main sort of 3 different regions, sort of the CA1, CA3, and then a gyrus. And just here showing you that we're really getting sort of a agreement between our data and their classification, which is very comparable to sort of just the internal agreement that the dataset itself has. So the correlation between the taxonomy to the taxonomy, right, sort of between different cells is very similar to what we're getting from sort of MERFISH to the taxonomy. So we're kind of in the end, right, the cell types that we find are indeed the cell types that sort of match what's happening there. Okay, and what was really interesting though is that we started to look not at those high-level types, CA1, CA3, and DG. We really tried to delve down into those sort of really small subtypes that the LN people sort of defined, right? And I'm showing you here two examples, both for the CA1 region, sorry, and the CA3 region, right? And what was really interesting to see is that those sort of specific neuronal subtypes are really sort of organized in space, right, from sort of side to side. So here the sort of subtype 10 is sort of starting, and then 9 and 2 and 5, so right, and sort of you can really see the progression until in the end, sort of here in sort of the in the right side, right, it's more sort of, you know, there is a higher frequency of sort of type 8 and type 6, right, and same thing for the sort of C3 region, right, really arguing that it's not even always clear if that segmentation, if those types are really distinct, or are we just looking at some continuum that just by the way they build the taxonomy, they sort of decided to break it down into specific sort of bins. What was really interesting to see is that really knowing space, right, knowing the spatial position of those cells really can explain a lot of the variance, right? So in other words, if I look at all neural, like CA1 neurons in just a single cell RNA-seq measurement, I will get a really high variance, right? I'll get a pretty big blob. And I'll say, well, cells are very heterogeneous, right? There's a variability among CA1 neurons. But really that variability is just a function of where they are in the tissue, right? Really arguing there's some sort of interesting kind of organization and functional state for those cells, right? It's not just sort of uncontrolled variability, it's really functional. And by looking at space, we can really see that happening, right? So another way to sort of think about this is really to ask, is knowing the position tell you about the transcription? And it seemed to be the case, although interesting, it's not really, the converse is not really true. So again, looking at those two regions, right, let's look at sort of the CA1 neurons, and this is frequency of neighbors. So for each one of those pairs of possible types, we're asking how many of your neighbors are, right, sort of from the other type. So this is sort of our measure of sort of co-localization, if you will, accord between two types, and just the gene expression correlation on the y-axis, right? So if you are co-localized, right, if a lot of your neighbors are shared, you will have very high gene expression correlation, right? So things that are next to each other really share similar sort of gene expression signature, but that's not necessarily the case the other way around. So you could have a really high expression correlation, but not have sort of co-localized, right? So things that are co-localized share transcriptional similarity, but it's not necessarily true of the converse, and the same thing is happening in the CA3 region. So this is when we looked at subtypes sort of within the same type, so subtypes of CA1 or subtype of CA3. Similar patterns also emerge when we look at sort of across different types. So we look at the between CA1 neuron and the SST interneurons. So it was really interesting to see that if we look at sort of neighbors, a subset of those SST neurons, right, sort of subset, right, 12, 20, 19, and 28, really are sort of seem co-localized with the CA1 neuron, and I'm just showing it to you, sort of I'm marking all of those subtypes in purple, right, and sort of all the CA1 in green and all the CA3 here in cyan, and you can see where they are here in the map in the bottom left, right? So if you are co-localized, right, you really have high gene expression correlation, right? But you can also have other sort of examples where you still have correlation, but you're not co-localized, right? So again, sort of being co-localized argue that you have sort of shared transcriptional signature, but the converse is not necessarily true. Okay, and finally, we sort of looked at that sample and tried to identify sort of spatially differential gene expression in genes. So we said, okay, within a specific subtype, right? So if I now look at sort of CA1, SP1, right, only for that specific cell type, right? What is the expression of one of those genes, for example, tox gene, is it sort of uniform, or does it have specific sort of differences in its spatial pattern? And we found lots of those. So this is just sort of 4 examples of, right? So it's always a cell type and a gene, right? So the tox gene in the CA1, SP1 subtype and so on. And sort of from the 80 or so genes that we looked at, basically we found roughly 40 of them showed some spatial differentiation. Those are genes that were not used to call cell type. Those are sort of non-marker genes that we had in our sample. So both sort of a lot of different, it was sort of basically a lot of the cell types had that pattern, and also a lot of the genes sort of showed that pattern as well. Okay, so trying to sort of wrap up, really we're sort of, I started telling you, the first story I told you guys was about sort of really trying to think of mechanistic view, right? Another mechanistic prism where we can sort of look at the cell state space and variability among cells through sort of the lens of a specific kinetic model, right? Of sort of single cell parameter fitting. I also showed you some data arguing that gene expression is really not that noisy, right? In our, at least in this system, right? It's pretty close to sort of Poisson limit. right? So basically now, if we account for a sort of shared component of variance between things, right, the gene-specific thing is very close to its limit, to its Poisson limit. And finally, sort of, I made an argument here that really a lot of that cell states type information, right, which is really just correlated gene expression and variability mentioned, is really reflecting underlying principle of tissue spatial organization, right, both in the sort of how subtype, right, can sort of spatially organize, but also in terms of the differential gene expression, right? So the variability of fragile in astrocytes really seem to be not random, right, in the sense, or at least not uniform in its randomness, right? There are definitely some areas that have higher expression and some areas that have lower expression, kind of arguing there's some underlying biology that we sort of still need to discover, right? Right? So, really, we need sort of the point I was lacking here is that we need to look at cells in multiple ways, which I think really resonate well with sort of the multi-omic theme that I really like how spatial biology is sort of trying to leverage on. But I kind of went saying, okay, we can also do multi-omics in really lots of different ways, right? We can really just think of them in an abstract way. Can we look at cells in multiple views, right? They're sort of, and kind of a new view we're trying to bring to the thing is really try to also use the underlying mechanism, right? At least in cases that we know what's happening there, or we think we know what's happening there, calcium signaling, can we actually use that to get a better sense of what's happening in each individual cells? But really the sort of final part of the talk really sort of argue that a lot of those views, right, will benefit from doing that with a spatial context, right? So we can build those layers of spatial information, and it will tell us a lot about really what's happening in cell state space. Excellent. And with that, I want to just acknowledge the people who actually did the work. So sort of Zach and Rob did all the sort of measurement of MERFISH for the TBI section, and Rob did all the sort of calcium and imaging. Russell from Xiao Zhang lab did all the work for the computational algorithm that we did for the segmentation, and Adam and his student Xiao Zhang are sort of helping us now with the single cell parameter fitting in the context of sort of joint gene expression data. Excellent. And with that, I'll be happy to take any questions.
Roy, this was a great presentation. So I think there are a few questions on the chat. Do you want me to read them, or can you read it?
Either way, let me open the chat. It was closed so far.
Mm-hmm.
How do I get the chat? Oh, here it is. Okay, so I'm answering the question, is the parameter fitting done independent for each cell? If so, how does fitting adjust for technical biological noise? So in the original version that I showed, so in the data that I showed you, yes, we have been fitting the parameters individually for every cell, and we're starting de novo every time. I would argue that basically already accounts for sort of the biological noise, which is really what we're after, right? It's the variability in that sample. What we're doing now with Adam McLean's lab is we're actually trying to be better at sort of this parameter fitting, because we can say, well, if I also know gene expression among cells, if we can build sort of a neighborhood graph on gene expression, can we actually sort of use your neighbors as a way to sort of start your prior, right? So rather than starting with sort of this uninformed prior, we can actually do that, right, by sort of saying, well, you're kind of similar in your expression to those 10 cells, and those 10 cells have those sort of parameter distribution, so we can do that. And that actually really speeds up things a lot, right? If you think of it in a limit of lots of cells, right, in the end, you just need to take a cell, find its neighbors, and immediately you get a prior that's actually very close to what the final posterior in that cell will be, right? Because it's sort of very informative prior. Hopefully that answers the question. And then I guess another question is about our sort of pixel level. How many transcripts does each pixel need to contain this procedure to work? So I'm not sure the final answer. We only did it so far on sort of this hippocampus tissue that I showed you. In this case, we had 80-ish, 83, I believe, sort of marker genes that we had, and another set of 80-something genes related to sort of the underlying biology. In this case, it's sort of a traumatic. There were genes that were expecting to respond from traumatic brain injury. So sort of, we had only 80 or so genes. Conceptually, I think it could work with sort of lower numbers as well, but we haven't actually done the work of sort of trying to, chunk it down and see how low can we go. I think, did I miss? Oh, how do you solve the local versus global optimization problem? Sort of the procedures we're doing, like we don't know, right? So that's a big problem in any sort of large scale optimization, right? There's no guarantee that we are in necessarily sort of global minima. The only thing we did is that we did fit for every cells. We fit it so the whole parameter sort of twice, right? And completely independent fitting run. And we sort of, if the two runs did not converge on similar parameter distribution, we sort of tossed it away. We did not include that cells in our cell type. So I cannot guarantee you that it's global. That's a really tough question, but I can basically say that we got to the same place twice. So if it's local, it's basically a big local pick, right? It's not some tiny thing that we get stuck on. Okay. Am I missing something else? Okay. Gene correlation in your y-axis should depend on what genes you're using. Yeah, so in this case, we're using, so there are two sort of steps here. So we had to split that I was describing. We had the marker genes and we have sort of the non-marker genes. In this case, we sort of define types based on the marker genes. So in both cases, sort of go in this sort of analysis and in a gene specific, we define the type based on those 80 marker genes, and then all the rest of the analysis was done on the other set of 80 genes. So there are genes that are chosen to basically because we expect some differential response in traumatic brain injury in warning the neurons about this, right? So that's sort of differential gene expression is sort of not part of the sort of cell type markers, right? There are specific, there are some other sort of biological sort of genes that are, respond now. Yeah, hopefully that answers the question.
Great. So, yeah, if you have any questions, please, you can unmute yourself or post it on the chat box. And in the meantime, I can probably ask one question. So these MERFISH data sets are 3D or just a few sections? How many sections do we have, myself, in the Z?
So currently so I showed you either cell culture or a single section. So nothing is 3D here. I mean, it's 3D in the sense that we're still imaging, even in a section, it's like a 15 micron section. So we're still taking a Z stack. And we're sort of, right, but it's not, so it's 3D in that sense, but the data is like a 2D 15, it's a 15 micron section. So it's pretty thin.
I see. And can you extend this pixel-based approach to voxel-based approach? And how would that work?
Oh, yeah, I mean, pixel is just a construct of our imagination, right? So I can always take the 4 pixels in a bin and call them, I think we even did that here. I think our pixel in our analysis is a bit bigger than our actual camera pixel, right? Sort of the first step that I didn't go for all the details, we're actually not using, right, the RNA comes in discrete spots, right? So we're actually doing some sort of local density estimate, right? So that already gives us some spatial scale. There is no point going sort of under it, right? And those are really determining our sort of effective pixel size, much more than the original sort of camera pixel, right? Because we can't do that if the sort of genes are too, yeah, if I have sort of dots that are too far away, right? I won't know anything about that, right? So I do need to have a good spatial measurement of gene expression.
So you use binning basically to address the problem, pixel binning, right?
Yeah, yeah. I mean, we're adjusting our sort of pixel size that we do all the computation on to this smoothing that we're using to estimate the density.
Oh, got it.
Right. So we have a kernel density. Well, we're doing it in a KNN approach, but it's equivalent, right? It's sort of we have some local density estimate, right, that we need for every area, and there's no point looking at pixels that are smaller than that local density, right? Because they'll basically be the same. So that's kind of determine our.
Okay. There's one more question.
So the data points, it's this one time series of calcium signaling. So I mean, there are 300 or so data points, but it's really time series data, right? So the 300 points are kind of just the DT that we're imaging, and we're just imaging fast enough that we can actually capture sort of what's going on there. Maybe kind of similar to the spatial problem, right? To make our fitting work a bit better, as I kind of showed you with those red dots along the line, we did have to sort of smooth a little bit calcium signaling, right, to sort of account for some of that, because otherwise the sort of some of those sort of spiky data kind of really threw off our parameter fitting. So we did sort of have to do some smoothing over the original data to really capture sort of the main response. And mostly it's because, and the justification for that is, again, our model does not have this ability to create fast spikes, right? That's how we chose our model. So there's no point asking the model to fit something that it sort of by definition cannot do, right? So we kind of adjusted our smoothing level to basically the sort of ability of our model to change, like how fast the model itself can actually change. So there's no steady state assumption here, right? We're looking at a time series data, right? So there's all, so basal, you add the ligand ATP, and then sort of it's all time series data all the way to the end. So we're not fitting to a single value of the cell, we're just fitting the time series, kind of a least square, if you will.
Great. And if in the meantime, I can then add one more question. By the way, Dr. Fan is in a committee meeting, so he cannot be here now, but we'll continue until finish, unfortunately. So the other question that I'm curious is, so you move from live imaging to fixed, right? And that actually interface, have you zoomed in a little bit more, maybe did a little slow fix or fast fix and played with that dynamics, right? Right at point of fixation. So because previously you have calcium dynamics, and after you have snapshots, right? So that time point is actually maybe immediately affecting, of course, the molecular distributions, right? But I'm not sure if you can also do a slow fix or whatnot to play with that also dynamics, right?
Oh, slow, we haven't even thought of slow fixation. So that's an interesting point. I'll think about it. But the reason we're not worried about it too much, again, is because of this sort of time scale of calcium signaling. The whole thing is 10 minutes. So I don't really think that a lot of mRNAs are really changing during this time. So, right, there's just not enough time for them to happen. When we do similar things on other pathways, sort of MEP kinase and NF-κB pathway, there we're much more concerned. And I'm not calling those genes necessarily the sort of state of the cell that caused, right? So here, sort of a causation is kind of nice, right? We have central dogma, and we basically say there's not enough time for the feedback to happen, right? So the RNA affect the protein that affect the signaling, and it's too fast for it to go back, right? But in many other systems, we can't do that, right? For sort of NF-κB, we image cells for a few hours, and it's actually part of the signaling pathway of NF-κB itself is the sort of gene expression response of the negative feedback regulator that sort of drag NF-κB out of the nucleus. So in many other systems, we can't play this game, which is one of the reasons we sort of like calcium here, right? It's fast, we fix it, and I don't have to worry too much about that, right? So hopefully that.
Yeah, it makes sense, totally. I was just curious about this experiment before, so. So any other questions? If not, let's thank to Rai again. I think there's this reaction clapping. I'm just going to use it. And
thank you very much, guys,
for this great talk. So next week we will continue our seminar series, and you can check more details on the Spatial Omics website and spatialomics.net. If you haven't seen it, please go and check out. And we'll be uploading these talks to that website as well. And you may want to access from that website. And other than that, I think next week we'll be seeing you guys hopefully on October 4th. And with this, I think we are concluding today. And have a nice weekend.
Excellent.